So let's start with the Alpha-proteobacteria. This is a phylogenetic tree that just shows a few of them. This is a hugely disparate group of creatures in terms of phenotype. Rhodobacter is a photostroph. Paracoccus is a nitrogen cycler. Rhizobium, a plant symbiont. Agrobacterium, also a plant symbiont. Bartonella, an animal pathogen. Hyphomicrobium is a filament. Methylobacter, these things oxidize methane for a living. Some nitrogen cyclers, and here's Rhodopseudomonas, purple photosynthesizers. Caulobacter, you guys have heard of Caulobacter before, has stalks. Rickettsia, an obligate intracellular parasite. Acetobacter, an organism that makes acetic acid for a living. So there are phenotypes all over the board here. Autotrophs, heterotrophs. Amongst both, there are organisms that use a wide range of materials to make energy and fixed carbon where they do. All kinds of lifestyles in here. Huge number of organisms in this group. Those that are autotrophic, fixed carbon using the Calvin cycle. Most of them have lipopolysaccharide except for Sphingomonas. There's another group of organisms that does that. They use sphingolipids. These are nitrogen-containing membrane lipids, such as those found in your central nervous system. Many of these organisms, not all of them, but many of them have appendages. You know of Caulobacter, which has a stalk. Also Rhodomicrobium, which many of you are seeing in your purple non-sulfur tubes in lab, of these organisms with stalks or filaments connecting them. These things are abundant in all phenotypes and all types. So who are they? The purple non-sulfur phototrophs are predominantly in this group. There are a few outside the group, but most purple non-sulfur bacteria are in this group. And so here's a Rhodomicrobium vanillii. And this, how many of you have seen this in your stuff in lab? Cool. Notice that it divides by budding. Here's the mother cell of this one. Here's a stalk, and the new cell forms as a bud at the other end of the stalk. Caulobacter is in this group. Caulobacter is the model system for studying chromosome division in bacteria because the life cycle is so neatly separated. And you can physically separate these two kinds of cells and create synchronous cultures. And so imagine taking a culture of Caulobacter. These things are attached to the surface, and these things are free swimming. So if you take a culture of Caulobacter, gently pour the supernatant off. The stuff that's stuck to the glass is all this. And then you add liquid media to it, let it sit for 5 minutes, and then pour it off again. All you get are newly released swarmer cells, all of the exact same part of the life cycle. And so you can follow them, their progression through their life cycle. It's a really, really easy thing to do. So the chromatin division is nicely separated from cell division in this organism. It's a great model study to study, great model system to study what amounts to mitosis in bacteria. The nitrogen-fixing plant symbionts are in this group. Agrobacterium. Most of you probably work with Agrobacterium in the undergraduate general micro lab. This is a plant symbiont. I guess you could consider it a parasite. It causes crown gall disease. But also in this group are the more symbiotic nitrogen fixers, things like Rhizobium, that form nodules in leguminous plants. An interesting thing about this organism is the use of a globin. like hemoglobin in the plants that modulate transport of oxygen to the organisms. Because these things fix nitrogen and are also aerobes, they have the same trouble we talked about earlier about how oxygen inhibits nitrogenase. And so they have to, the bacteria are fed oxygen not in solution, but bound to like hemoglobin. This is a great model system in which the organism has a large chromosome and 6 smaller chromosomes. Most of the genes on those smaller chromosomes are non-essential, but there are some housekeeping genes on that too. And so they really are secondary chromosomes in this organism. There's a whole group of obligate intracellular parasites in this group, in the Alpha-proteobacteria. Rickettsia, Bartonella, Wolbachia, this sort of thing. Also the mitochondria. And so mitochondria are members of the Alpha-proteobacteria. Wolbachia is a great case. Wolbachia is an insect pathogen. A pretty significant fraction of insects are infected with Wolbachia. And in most cases, it does them little harm. Wolbachia is most commonly transmitted by females during egg laying. And so the Wolbachia is in the ovaries. And this is showing, this is an egg of a wasp. And here you have these little spots are Wolbachia cells stuck to the egg. And so when the eggs are laying, the Wolbachia is transmitted to those eggs. Now this creates a really interesting evolutionary paradox for Wolbachia. If this egg is a female egg, then that infection will result in an adult female wasp that can in turn transmit the Wolbachia to its offspring, right? But what if it's a male egg? Wolbachia is not capable of infecting sperm and having that transmitted. That means that if this is a male egg, these Wolbachia cells are an evolutionary dead end. They can infect that male, but it will go no further than that. And so half of the Wolbachia in that mother's oviduct would be destined for dead ends. And so it's very common for Wolbachia and for other members of this group to be what are called sun killers. Where if this is a male egg, the Wolbachia will kill it. The females then will recycle that energy and create more eggs, half of which will be female, right? And the result is more female egg production. Better transmission to the next generation by Wolbachia, but that skews the sex ratio of the host. And in many cases, these insect sex ratios can be very heavily modified to the point where the females have difficulty finding males for reproduction and resort to parthenogenesis instead. And so you see feminization of insect populations and even parthenogenesis in insect populations as a result of Wolbachia infection. There's some evidence also that that Wolbachia infection may prevent the transmission of malaria. And so people are using, are working to use Wolbachia to infect mosquitoes to fight malaria.